This webinar is co-hosted by the National Health Council and the Duke-Margolis Center for Health Policy, and is part of our joint educational series on an introduction to real-world data and real-world evidence, a virtual training series for the patient community. Next slide, Sarah. Before we get started, I would like to just go over a few housekeeping items. The webinar will be recorded and then also archived on the NHC website. And you can find it at the same location as where you registered. All participants are automatically muted when you join the webinar, and we're going to have time at the end for Q&A. And so if you could please submit any questions through the Q&A section, we'll try to be responding to some of those as we go, and then have them at the end for discussion. Next slide. So this webinar is part of the PCORI funded training series for the patient community on real world data and real world evidence. And we'd like to extend our thanks to the advisory board members that are listed here on this slide. Next slide. And then here's just a list of the previous webinars in the series. And then the next one that is on the series, it's on the sources of real world data, patient-reported outcomes collected during routine healthcare delivery, and it's going to be on April 23rd. Excuse me. If you're interested, please head over to the NHC website to register or to view previous webinars. Next slide. And another thing I wanted to talk about is that we're now offering scholarships to members of the patient community that attend our webinars. So if you are a patient or a patient advocate, you can go to our website and apply for a scholarship, and if you've attended at least one webinar in this series. So be sure to check that out. Okay, so I want to just introduce you to the speakers today. So my name is Gillian Schmidler. I'm a professor of population health sciences at Duke University, and then also deputy director of the Duke-Margolis Center for Health Policy. I'm going to be joined by two speakers today. Robert McBurney and Kelly Myers. Robert McBurney is the Chief Research Officer and former CEO of the Accelerated Cure Project for Multiple Sclerosis, or ACP. ACP is a nonprofit organization accelerating multiple sclerosis research to improve the healthcare and quality of life of people affected with MS. And he's also co-PI on the I Conquer MS People-Powered Research Network, which I think we'll hear some more about. He has over 40 years of biomedical research and management experience, from conducting medical research as an academic neuroscientist in Australia, the United Kingdom, and the US, to leading biotech companies in the UK and the USA. And then second, we're going to have Kelly Myers, who's a healthcare IT entrepreneur with over 32 years experience. He's held senior level positions in Fortune 100 pharmaceutical companies and has founded and led successful healthcare analytics startups. He's the Chief Technology Officer to the familiar Hypercholesterolemia, our FH Foundation, and leads the Find FF initiative. And his responsibilities include leading the development of the Find FF algorithm and the data mining platform. So I'm really looking forward to hearing from both of them, and we're going to make sure we have about 45 minutes together. We'll hear from both of them and then have some time at the end for discussion. So please make sure you think of the questions, share them in the Q&A, and turn it over to Robert.
Thank you so much, Gillian. I really appreciate the opportunity to participate in this webinar, and thank the National Health Council and the Duke-Margolis Center for their work in this area of educating people about real-world data. I'd like to talk today about clinical data registries for real-world data with some examples from multiple sclerosis. And I hope that what I talk about today is something that will inspire people to participate in registries. Next slide, please. I'll just start with some words from the website of the Multiple Sclerosis International Federation. As you know, multiple sclerosis is a chronic degenerative disorder of the nervous system that affects people all over the world in many different ways. There is no uniform patient experience of multiple sclerosis. It's highly variable. and it's generally diagnosed in early adulthood. There are some pediatric patients, but it is at the moment a lifelong chronic degenerative disorder. The Multiple Sclerosis International Federation goes on to say that MS registries are an advance in MS research that do two things. They bring people closer as a global MS community. I'll talk a little bit about that, and bring people closer to ending MS forever. And what I'm going to talk about today is one of the two general ways that registries gain their data content. And the way I'm going to talk about is the collection of clinical data, primarily from doctors. In other webinars and in the webinar next week, in April, you'll hear more about patient-reported outcomes, which is a very, very important component of registries as well. But for now, I'm just going to talk about clinical data registries. Next slide, please. So a simple registry has a number of features. And by simple, it doesn't mean that it's simple to create, but it's maybe the simplest version of a registry. And so in an encounter between a clinician and a patient, data gets entered primarily by the clinician into the electronic medical record. Data can come from the clinician, or it can come from other sources like magnetic resonance images or lab results or other sources. And all of that data gets into the electronic medical record, where it is linked to the patient identity for the purpose of care. And so that's the function of the electronic medical record, is for care and other activities such as billing. But primarily, the data is used for care. A registry generally contains data that is anonymized. In other words, the many things that can identify the data with a person is removed as the data is transferred to a registry. And often these registries are third-party registries, and we can talk about that maybe in the question time. And there are 18 elements that have been identified to be removed from the data in order to make it anonymous. And so that's really the picture of a simple registry, which contains anonymous data not linked to anything other than a code which it links all of the data from one individual together, but that code doesn't tell you anything about the identity of the individual. The next slide, please. Of course, there are more complicated registries, and I give this example here because it's a very relevant example in the United States at the moment. And so in addition to the simple registry containing data from an electronic medical record that is anonymized, there are many initiatives that also hold biological samples like blood or tissue samples, and they are held in a biorepository, and they are usually consented by the patient to be held in that biorepository. And then there is a link using the same code that is given to the data. There's a link to those samples through that code. So the samples are also anonymized in the repository. Now, I mention this type of repository because many of you may have heard through your healthcare providers or your hospital systems of an initiative in the United States called All of Us. And this is an initiative that was initiated by the National Institutes of Health, where this type of registry linked to biological samples is going to be created, is being created, and will be available for all sorts of research into health conditions for the United States. It's a tremendous potential resource for accelerating research in many, many different medical conditions. Next slide, please. Now, of course, I've talked about just up till now the idea that a clinical site data gets into a third-party registry. And the example I gave before of All of Us is a registry that is contributed to by many different clinical sites. And it comes along with the biorepository as well. And so the idea that multiple clinical sites can contribute data to a registry is really important. And the reason it's important is because not every clinical site, particularly the sites associated with rare diseases, will have sufficient data on people with rare diseases to enable adequate research. And so it's really important that data from multiple clinical sites is brought together into a registry to allow, in an anonymized fashion, to allow research to be undertaken to improve outcomes for people, particularly with rare conditions. And one example of this is PCORNet. PCORNet is the National Patient-Centered Clinical Research Network that was established in 2014 with funding from the Patient-Centered Outcomes Research Institute that was created by the Affordable Care Act. And this today is a tremendous resource for healthcare research. It involved 12 hubs, sort of super systems. And underneath those 12 hubs, there are 80 healthcare systems across the United States that are contributing to the PCORNet data. And that registry currently contains data on 70 million Americans. So this has been a tremendous achievement in the past 6 or so years to create this national resource. Next slide, please. So I just mentioned a few key considerations before I talk about MS, multiple sclerosis specifically. People whose data get into registries sometimes don't consent because once the data is de-identified, it's not considered personal health information and therefore does not require generally require ethical approval or individual patient consent. Not every situation is like that. Sometimes there's an overall or blanket consent from patients for particular registries to be created. And then there are even examples where every data element in a registry is under the control of the person who contributed that data. And so for particular studies, people can identify whether this data point will be used or that data point can be used. And that's giving people with health conditions ultimate control over every data element in a registry. This is not the case in PCORNet, nor in All of Us, but certain data registries do have that capability. And I think for some purposes, it's tremendously comforting to patients to know that their data is under their control. There are a variety of things to do with governance of registries that are important. Does the governance of the registry include clinicians, researchers, patients, care support partners, Or does the governance only include researchers, for example? And I think that when looking at the organization that set up the registry, it's very important to look at the governance of the registry, because that assures that the data, the approval of data and biosamples for specific research studies really comes from a broad spectrum of stakeholders involved in that particular registry or in that particular medical research area. And I think it's very important from a governance point of view that registries are associated with open science, which means that if I take data from the registry with approval of the governance, or biosamples from the registry with approval of the governance, that I make a commitment as a researcher to share that data back with other researchers, not necessarily just through publication, because publication is usually associated with research that is successful in one way or another. But I think the sharing of data from research studies is a very important component of open science, and the governance of a registry should be interested in ensuring that the registry data is used in an open way for the benefit of all. And then there's a very important component of registries, which is sustainability. So many registries are set up to go forever, to follow people with health conditions for a very long period of time. Registries don't come without cost. And so I think it's very important to understand, if you're looking at contributing to a registry in a proactive way, what the sustainability is for that registry. Next slide, please. So I just give you some example from multiple sclerosis. Multiple sclerosis is very fortunate to have many, many national registries and some registries that pull together data from multiple registries. And so, for example, in Europe, there are individual registries associated with specific countries, such as the Danish Multiple Sclerosis Registry, the Swedish Registry, and the Italian Multiple Sclerosis Register. And these are in some cases funded by a government, like the Swedish Registry, and in other cases funded by foundations, like the Italian Multiple Sclerosis Register. These are particularly important because they have a strong element of sustainability associated with them. In addition to that, there's an organization which began in Australia, my home country, called MSBase, which is a way that clinical sites can directly enter data into a big registry, and also some of the international registries also allow their data to be, their anonymized data to be transferred into MSBase. MSBase today has a registry that represents about 80,000 people with MS. And it's a tremendous resource, and I'll come to that in a moment. And just recently, the last 6 years, something called the Big MS Data Network was formed that combines the registries of a number of governments or a number of countries, as well as parts of MSBase. And it currently represents over 200,000 people with MS. And so the national registries and the sort of supranational registries are very important because I mentioned before, multiple sclerosis is a very heterogeneous medical condition, and it comes in different forms with different symptoms and different rates of progression. And to really understand it, we need big data. Just down below, I have a couple of other things represented. 2 industry-sponsored registries called MS Link and MS Paths. which have just got off the ground in the last few years, MS Link most recently, and also the Accelerated Cure Project repository, which contains both data and biosamples collected from 10 clinical MS clinics across the United States. Next slide, please. And I'd just like to finish with giving you some idea of how registries enable MS research worldwide. And this would be just an example in MS. There are many, many examples in other fields. So a registry like MSBase has published many papers. It's a resource for researchers worldwide, going back to the statement that was made on the Multiple Sclerosis International Federation website. And it has brought people together, definitely. And for example, to study the effect of a disease-modifying therapy on disability over a 15-year period requires a real attention to data, because currently there are about 20 disease-modifying therapies that have been approved by regulatory authorities worldwide. And people with all sorts of levels of disability have been treated with these therapies. And it's only a big database that can really study the effect of disability. And it's also only a real big database that can look to see whether there are early clinical markers that can identify those people with aggressive multiple sclerosis. And there was a wonderful paper published last year. And I've gone from showing none of the authors to just a small number of the authors in the second one. But for the third example here, which is to do with the incidence of pregnancy and disease-modifying therapy exposure and what the trends are, I've shown both the title of the paper and all of the authors. And if you look extremely closely, you'll see that there are 40 different identifications of where those authors came from. And so this is also what a registry can do. It can not only bring together people for the contribution of data, but it also brings together researchers for tackling really difficult studies. Anyway, so that's really the story about clinical data registries that I wanted to talk about. And the next slide just summarizes. The conclusion: registries are important research for accelerating research in all diseases, and especially for rare diseases where often no clinical site has enough data. And I think Kelly's going to pick up on that in a moment. I think the governance of a registry is of critical importance to ensure that the data and biosamples are used ethically and for the ultimate benefit of patients. And as I mentioned, sustainability is a key consideration. And I really encourage people to do their homework about registries they might be invited to participate in and participate. And thank you very much.
Wonderful, thank you. So we'll go to our next talk with Dr. Myers and then save our questions for the end. There are a few questions that have started popping up in the Q&A, and I'll let you, Robert, you might want to take a look at those, and then we'll make sure we have time at the end. Okay, Dr. Myers. Yeah, there you go.
On behalf of the FH Foundation, thank you very much to the National Health Council and Duke-Margolis for this opportunity. Familial hypercholesterolemia affects 1 in 250 individuals worldwide. It's an autosomal dominant condition. So once an individual is identified with FH, there's a 50-50 chance that all first-degree relatives will have FH as well. That's significant because these individuals are exposed to lifelong, very high lipid levels. And if left untreated, can start having heart attacks in their early to mid 30s. So that's all the bad news with FH. The good news is if identified early enough, it's highly addressable. A lot of individuals will respond easily to generic medications. So the FH Foundation was founded 10 years ago by Catherine Wildman to address the challenge of the fact that the overwhelming majority of individuals worldwide with FH are undiagnosed. 10 years ago, when she started the foundation, it was estimated that about 1% of individuals with FH were diagnosed. So the FH Foundation is very much about finding a way to get these folks identified and brought into care. Next slide, please. So in that, the method by which the FH Foundation tackles these challenges is to bring data, collaborate with FH experts from around the world, and also highlight those individuals impacted by FH and their experience, and bring all three of these things together to fight this challenge. Next slide. So from a data perspective, we have developed the Find FH initiative, and this initiative has been now in its 7th year and has utilized a big data-based approach to help expedite the identification, screening, and diagnosis of these individuals. And as you can see here with these objectives, we sought to find folks early and get them brought into care. These types of initiatives require a lot of resources, and we're happy to acknowledge that Amgen is a foundation level sponsor of this program and has been since the beginning of the program. Next slide. And one more click. So the FH Foundation set about developing this initiative before there was a diagnosis code for FH. Next slide. So we had to go out and identify individuals to train this artificial intelligence, machine learning based model. And we had to do it in a very manual process by partnering with 4 institutions, Penn, Stanford, Geisinger, and Ohio State, to identify about 1,000 true positive individuals with FH and about 40,000 individuals that were believed not to have FH, and sent them through a machine learning process by which the machine looks at that individual's history and identifies characteristics that are common to those individuals that eventually end up with a diagnosis of FH. Some of the characteristics that the model looks at are things like cholesterol, or how recalcitrant their cholesterol is no matter what therapies they're on, the age in which the individual is, and then it also looks at a number of non-clinical aspects of how many different agents has an individual been on, or how many encounters they have with the health system. All of these things become a vapor trail, if you will, a pattern that the computer picks up on by which to get these folks identified. Next slide. And so when going through this process and working with the data from these institutions, we were successful in building a model that has a positive predictive value of about 80%. So with an overall prevalence of 1 in 220 or 1 in 250, to find 8 individuals with FH, if you were doing population-based screening, you'd need to screen about 1,800 or 1,900 individuals. With the Find FH model, to find those same 8 individuals, the model will identify 10 individuals and be right on 8 of them. These studies have been validated both in a publication in Lancet Digital Health as well as Nature Digital Medicine, and we're happy to get you the citations for those, if you will. So once we built the model from these 4 institutions, we further evolved the model with a national data set. Next slide. And this national data set was built with a combination of healthcare encounter data from all payer types and all institutions. We have over 300 million individuals with at least one healthcare encounter in the database, and lab result data on over 100 million individuals. which makes this a very unique data set. And when we evolved the model that was developed in the 4 institutions and applied it to the national data set, we identified over 1.3 million yet to be diagnosed individuals with FH in the national data. And you can see a relative heat map here of where the model thinks those folks are. And again, the model is a screening tool, it's not a diagnosis. And the model's goal is to identify an individual that a clinician should evaluate. Once we applied the model to the national data, we then developed a process by which the FH Foundation can collaborate with local health systems in identifying individuals within their data. The next slide. And in this process involves getting a data extract from their EHR. We secure that data as a limited data set to address some of the questions that Robert brought up, and that the individual's identity is blinded to the FH Foundation, but the health system keeps the key, if you will. So the individual's a number to us, and we apply the model and return the results back to the institution. And then the FH Foundation partners with the institution on a reach out first to the impacted individual's primary care physician, and then most institutions that we're partnering with are triaging these individuals to a preventative cardiology clinic for their further evaluation, whether it be a clinical workup or genetic testing. And we're currently partnering with 6 health systems, and we'll show you those folks in a minute. And so the next slide. So in doing this process, everything that I've shown you up until now kind of addresses the technical aspects of it. But partnering with the institution to disseminate this information, that's the heavy lift. In that, make sure that primary care and specialty care are on board and understanding what this overall Find FH initiative can do, communicating with individuals on the importance of coming in and being evaluated within the specialty clinic area. All of these things are tools that have been developed by the FH Foundation to help this process going forward. Next click. And currently we're partnering with 6 different health systems on the implementation. In each of these health systems, we're at various stages of implementation. And you see here a mixture of both academic centers as well as community-based health centers. And we're having good impact as we're going forward. Next slide. And then just as a little further detail, the 2 papers that we published showed that the overall accuracy of the model, or the positive predictive value of the model, was about 80%, and being able to successfully identify the right individual about 8 times out of 10. Interestingly enough, the 2 times out of 10 that the model is incorrect, when they're brought in and evaluated for FH, about 90% of those individuals that don't have FH end up having their therapy changed or modified, because the model is picking up on some element of risk, even if it is not necessarily identified as FH. So that's the end of my prepared comments, and we look forward to the questions.
Great, thank you. Okay, so now we have a few minutes for some questions. I saw one coming in that I could start with, was asking about overcoming barriers of trust within healthcare systems for underrepresented minority or ethnic communities as it relates to expansion of real world evidence. And so didn't know what some strategies were that either of you are either using in your registries or aware of within the field.
I think this is a fabulous question, and a question that is really very important. Our ICONC-MS network, which is primarily patient-reported outcomes, is doing a great deal of work, and we've in this area because I think there is a lot of history associated with healthcare systems and data and underrepresented minorities. And I think there's a really significant effort that needs to be put into this area. And one of the most important reasons is that even with an adequate representation in clinical trials of underrepresented minorities, since the clinical trial as a whole needs the full population in order to determine a result. If let's say in a case that rarely happens, there were 13% of African Americans in that clinical trial. It's actually not possible to conclude anything because that 13% is not enough to make an independent conclusion about a subset. And so participating in registries where information can be gathered after maybe a drug is approved for the general population is a way of gaining information about underrepresented minorities in relationship to the effectiveness and safety of that therapy. And so this is overcoming these barriers are really important, and we have created a minority engagement partnership network in multiple sclerosis. We've also created a number of other initiatives, something we call the RIDE Council, which is inclusion, diversity, and equity in research. And I think we need more of these initiatives because I think that there is a significant barrier that is preventing us gaining the evidence that we need to ensure that treatments are safe and effective for populations that are generally underrepresented in clinical trials.
Wonderful, thanks. Another question. So I wanted to first of all encourage folks to ask questions in the Q&A, and I'll try to keep up to date in that. And so the another question is related to, I think, Robert, you mentioned the importance of the sustainability of the different registries. And I guess one question is, how do patients actually determine kind of a sustainability plan? Is that normally something with a registry that is able to be seen from the outside, or what kind of other methods do you suggest people use in order, because to make sure that the registries where they are helping to contribute data are going to be able to be around and useful for their different purposes?
I'll take a stab at that. Maybe Kelly can talk about specifics with regard to familial hypercholesterolemia, but I do think that in certain other countries worldwide, there has been a national commitment to registries for certain diseases, and I mentioned the Swedish MS Registry. And while national commitments to funding of governments is never guaranteed, I think it's very hard for such valuable information just to be terminated. And so I think in those cases, the national commitments are very important. I think that all of us represents a national commitment in the United States to a certain type of registry that can enable lots of advances in personalized medicine, for example.
Thanks, Robert. The FH Foundation also does maintain a national registry, and we seek support for that registry from a number of sources. One, Kat Davis Ahmed, who's in the audience today, heads our policy and outreach program. So there are things that we're doing to seek national support. We also receive industry support as well as support from individuals. The registry has been, the foundation is around for 10 years. The registry's been around for 8. And because we have 8 years of data now, there's a lot of questions that can be answered via that registry about the importance of care and what did care look like before the latest round of new therapies, what does care look like now, and what is the significance of the long-term close management of these individuals that, quite frankly, can only be really answered in a registry of this type.
Great. And another question that came in is about the challenges and opportunities of using EHR data. I know you've both spoken about getting data directly from clinicians, from the patients themselves. There's obviously data in the registries that's based on claims data, administrative data. What are some of the challenges in terms of the both the I guess quality and appropriateness of the different data sources for these types of registries?
Yeah. So, Robert, if you'd like, I'll take a first stab.
Yeah, you go first.
We're actually in an evolution of how we've done it. With the registry, we originally started the way a lot of registries do. There's 40 member sites within the Cascade FH registry, and the sites agreed on 100 data elements that would be extracted by a clinical coordinator and updated. So the registry was historically built under that framework, but we're starting to evolve because of the Find FH initiative. We're securing a large EHR data set for the Find.
Has Kelly frozen?
Yeah, he froze
Okay.
for me as well. So maybe if you want to jump in,
let me pick up on
Yeah.
that, because I think this is an excellent segue to the next webinar. So in multiple sclerosis, the symptoms and abilities that affect people with MS are often not captured in the electronic health record. And so the number one symptom experienced by people with MS is often fatigue, whereas the focus of the neurologist is often on various neurological functions and mobility, for example. And I think that one of the failings of clinical data registries is that they only have something that's in the electronic health record. And often, even though in an encounter between a physician and an MS patient, they may ask the question, so how have you been since the last time I saw you? And the patient will say, well, I've had tremendous fatigue. I haven't been sleeping well. I have a certain degree of anxiety, etc. Very, very little of that gets into the electronic record as actual data. And I think this is where the combination of patient-reported outcomes collected in a care setting in a formal way, combined with clinical data, can really enhance the registries. And so people who are coming to the next webinar will learn a lot about patient-reported outcomes being collected in the care setting. But I think that the clinical data alone is often not adequate to answer all of the questions that on topics that may matter to patients particularly.
Great, wonderful. And then Kelly, I see you're unfrozen. I didn't know if there was anything you wanted to add to that.
No, long story short, we're trying to evolve to a hybrid model. Capture automatically what you can capture automatically, and then have a small portion of the data, like the data Robert just described, that still need to be captured manually.
Great. Okay, well, I think we actually are at the end of our time. And so I do want to thank you both for joining us today. It was wonderful to hear updates on that. And also, as you mentioned, it really does lead into the next webinar that we are doing on April 23rd, focusing on the sources of real world evidence and the real world data with patient reported outcomes. And so I encourage folks to register for that and join there, and also check out on the website with additional recordings from this one and information about other webinars to be joining and upcoming events. Thanks everybody, and have a good rest of the day and a good weekend.